Hello and welcome back to this tutorial. Today I'm going to talk about alignment of COVID-19 DNA sequences and protein sequences, how to do it in Python using spike proteins of COVID-19. So I'll be focusing on the spike protein of COVID-19 as an example. We'll need to install Biopython and ClustalW software package. So Biopython is actually a library in Python. You can install it using bank pip install Biopython, and if you already have the library installed, it will tell you that the requirement is already satisfied. If not, it will directly install the package for you. If you'd like to install ClustalW, which you should actually in order to work on alignments, what you've got to do is to go to ClustalW software webpage by navigating to Google and writing down ClustalW download. It will directly navigate you to the download page of ClustalW, and you can download it for Windows, Linux or Mac. So this is the web page. You can either download it from here or click on the download tab in order to download the software. Once you download the ClustalW2 software, you can install it in a repository of your own inside your operating system files. So I installed it in program file 86 in ClustalW folder, and the file resides there. I'm going to show you later on how to use this path if you are not familiar on using the path in Python or any programming language. Once you do that, you can use global sequence alignment utility of ClustalW by importing the command line of the software. So you simply write down from bio.align applications or applications import ClustalW command line. So this is the class or the module that has the command line interface of ClustalW. There are so many functions and methods that are written for ClustalW alignment, and it will treat your DNA and protein sequences as strings to be iterated on later on. We need to have a file, a variable actually, to contain the unaligned DNA sequences that you have. I actually downloaded all of the protein sequences that I have for the spike protein in a file, a FASTA file called unaligned FASTA. So I'd recommend you to name your file this way in order to clearly understand the procedure behind it, and place the file next to or in the folder of your Jupyter Notebook or Python file. So this is the protein containing file. It contains a number of nearly identical protein sequences of the COVID-19 spike proteins that have been sequenced and published on NCBI. So in the Genbank you can find all of these sequences. You can track the Genbank IDs and nucleotide IDs and then download them. Or if you need them, just comment below and I'm going to send you the file. So once you have that variable containing the file, you can parse the DNA sequence and then use the ClustalW command line to read the file or parse the file. So this parsing is quite important. So to do so, you write down ClustalW command line, open parentheses, and in between string quotations you write down ClustalW2 in file equals in underscore file. The in underscore file is the same variable as we declared earlier, so that we can use it for the ClustalW application. So this software operates on files, and that file here is actually the one that we just opened, the FASTA file that we opened. We can perform alignment directly or perform assertions first. So let's do assertions, but before doing the assertion we can print out whether the ClustalW alignment is done or there is a problem. If there's no problem, it will show you this output, ClustalW2 in file equals unaligned.fasta. That's what it shows. There are two methods to read sequence outputs for alignments. One of them is called align.io.read. The second one is called align.io.parse. They both are under the bio module. I normally use the .read method, but you're welcome to use the other. To do assertions, you import OS and you write down ClustalW.exe is equal to the location of the file that you have. So remember, we had the location of the executable file of the ClustalW software in our repository in C drive. So you navigate to that one and copy the path. Then once you copied the path, you follow it by / or / ClustalW2.exe so that Biopython recognizes which file you would like to use. So this is just a navigation to navigate to the exe to be used. It's easier to use variables inside other variables. So we declare another variable called ClustalW command line so that we use the command line of the software in the ClustalW exe. So you write down ClustalW command line, note the camel casing equals to the command line, open parentheses, ClustalW exe, then in file equals to unaligned.fasta. So this activates the command line of the software to operate on the unaligned protein or DNA sequence inside the file, the FASTA file. So this exe will operate based on the command line call of the ClustalW process that we called it to work on the unaligned file. Now we do the assertion. You write down assert, then os.path.isFile, then show the ClustalW command line exe because it contains the software working on the file. And the assertion is for the ClustalW. If it works properly, it doesn't show anything. If it doesn't work, it tells you that ClustalW execute or executable does not exist, or is missing, or it's not found. So feel free to write whatever you need in whatever language you want here. Then we need to use two other methods, stdout and stderr. These are equal to the ClustalW command line. This is a method, so you have to remember using open close parentheses. Finally, use print statement to print out the ClustalW command line. This way you print out the output of the assertion. If it works here, it creates two files for you. One of them is the alignment file and the other one is the dendrogram. So we need to create the alignment first, then the dendrogram of the alignment. If you write everything properly and print out, you will get two files. One of them is the unaligned.aln, the other is .dnd. These two files are the alignment and the dendrogram for you, which is similar to phylogenetic tree, but using ASCII code. To show the output of the alignment, you have to generate the read method. So to do so, you write down from bio import align io, and using align io to operate on input output of the files, you write down clustal align equals, this is a variable by the way, equals align io dot read and open close parentheses. In between, write down unaligned.aln. ALN stands for alignment, so this is the aligned file that has been created by Biopython, and we have to tell the software that we use Clustal as the method of reading the alignment. Now we are able to print out the alignment, and this will show you the alignment just like any other software in the interpreter. Here we have Jupyter Notebook, and it will show you the output inside the Jupyter Notebook as it's shown here. So you can't see deletions, insertions, and gaps here because the sequences are similar. They are identical actually. Otherwise you could see gaps, deletions, and insertions every now and then. But for this example, the sequences are nearly 100% similar, except for a few of them that are not shown here, but I'll show you this in the dendrogram. So these two files are the alignment and the dendrogram that we created based on the alignment methods of Biopython. You can view them in Notebook. This is the alignment viewed in Notebook++ software. This is just similar to what we showed and what we've seen actually in Jupyter Notebook. Finally, to generate dendrogram or a tree output, use the following method. First, import philo, bio.philo. So you can write down from bio import philo, which is a module to generate phylogenetic trees, and declare a variable called tree and write down philo.read to read your tree, and the tree is saved in a file called nline.dnd. Use a method called newig for a method like newig. You can generate outputs of dendrograms. Then show the dendrogram in a draw ASCII method. You have to put the tree variable inside it, and this is the output of the tree. So you can see the similarities and clustering based on the tree output. There are some sequences that are quite similar and others that are not that much similar. So that's how you do it, and I hope you enjoyed it. Thank you for joining me in this tutorial. Until next time.